Hi, my name is Ryan. I'm a sophomore here at Bond Coors Memorial College of Nursing, and for my genomics presentation, I've researched the history of prenatal diagnosis and how we tend on improving this practice. So prenatal diagnosis means that it's just a diagnosis before birth, so it's how a doctor can see if there's any issues with your developing baby. According to the U.S. National Library of Medicine, approximately between 3 and 5% of pregnancies are complicated by birth defects or genetic disorders, with chromosomal abnormalities being present in approximately 1 in every 150 births. Prenatal detection has been offered for about 40 years now, first by amniocentesis, which was in the early 1970s, and additionally by chorionic villus sampling, which I'll refer to as CVS, in 1980s. However, CVS is an incredibly invasive procedure where fetal cells are extracted from the mother's abdomen very close to the fetus, and there's a low risk of miscarriage with these, approximately 1 in 1,000 for amniocentesis and 1 in every 500 for CVS. But all this has actually prompted a long quest to find a more precise, non-invasive prenatal test. All starting in 1893, where scientist Schmoller first suggested that nucleated fetal cells may be found in maternal circulation and that portions of the villus system of the placenta can become detached and pass into the mother's blood. However, direct analysis of these whole fetal cells from maternal circulation has been really challenging just given the scarcity of the cells in the mother's blood. It's about 1 out of every 10,000, all the way ranging to 1 in every million. And the focus has shifted from analysis to cell-free fetal DNA, which is found in concentrations about 25 times higher than that of nucleated blood cells extracted from similar volume of whole maternal blood. At 7 weeks gestation, the fetus shares bloodstream with the mother, and they start to excrete off some of their own cells, which then die and burst open. These are what we call the cell-free fetal DNA. And they start flowing around the mother's bloodstream, and they comprise about 2 to 10% of the mother's blood at that point. But the fetal DNA being available in the bloodstream does give us the rare opportunity to gather the DNA out of the maternal's blood instead of through the abdomen, which is close to the fetus, to detect the trisomies, trisomies are a condition where there's an extra copy of a chromosome that is present in a cell's nuclei, causing developmental abnormalities. So with the cell-free fetal DNA, we've been able to detect trisomy 21, which happens about 1 in 800 births, trisomy 18, 1 in every 7,500 births, and also trisomy 13, which is available in active in 1 in every 15,000 live births. With free cell fetal DNA, we've also been using it to screen for the Rh blood type of the fetus, as well as sex. However, both ultrasound and also maternal serum analysis are both still considered just screening procedures, and both still are required to be followed up by a CVS procedure if there's a screen positive case for actually definite diagnosis of a chromosomal abnormality in the fetus. So the cell-free DNA analysis is not a perfect process by any stretch of the imagination, and it still requires testing of fetal cells to confirm the diagnosis. For the future of non-invasive prenatal diagnosis, we need to be able to be more exact to avoid having to do the more invasive following diagnosis of the CVS testing in order to confirm the screening results. In order to do so, we have many researchers working on this, such as Dr. Boudet of Bayer College of Medicine. He's been working with his team to be able to develop a cell-based prenatal screening for many years, and he has had success with cell-based testing in a research setting. So in order to find and better detect those 1 in every 10,000 to 1 in every million fetal cells in the maternal bloodstream, Dr. Boudet is attempting a technique known as RareSight, which is microscopically scanning all the nucleated cells that in the sample, just to identify the ones that stain positively for a protein known as cytokeratin, which he is able to then use to determine which ones are the fetal cells. Another method being tested by Dr. Stein of Arcady Biotech has a different approach, and he's using antibodies that will only specifically attach to the fetal cells, and then he magnetically will pull them out of the maternal blood. So Dr. Stein highlighted why using whole fetal cells is so advantageous for these tests, and he was quoted because cell-based non-invasive prenatal diagnosis technologies offer intact fetal cells with access to complete fetal genome, which has also been uncontaminated by the maternal DNA. The possibilities of even detecting the smallest changes are only possible through using the full intact fetal cells. So the method by Dr. Stein has been successful in using nucleated fetal cells to detect subchromosomal abnormalities. So abnormalities not just with the entire chromosome, but actually inside in the individual parts, such as 31 MB translocated between chromosomes 4 and 8, and also 12.2 MB duplication on chromosome number 21. And they've also been able to prove these tests with using the CVS test just to confirm their accuracy. But similar to Dr. Bidet, he's saying that while the technique is working reasonably well in the lab setting, that their next step is trying to take it to the clinical setting, and they're projecting to try to have it on market in about 2 to 3 years. Also, researchers from Wayne State University School of Medicine also published their work last year showing that they could profile fetal trophoblast cells extracted from endocervical canal. It is in a technique similar to that used in the pap smear test at about 5-week gestation. In a challenge to proponents of the cfDNA testing, which is the cell-free DNA, fetal DNA testing, Wayne State University and also the 2 other colleagues have speculated that in the near future we will see cell-based non-invasive prenatal diagnosis taking over from cell-free non-invasive prenatal testing, and also in the long run it will replace all invasive prenatal diagnostics methods such as the CVS. So what would a more exact clinical approved non-invasive prenatal diagnosis really mean for us? Well, for starters, what is the ultimate goal for a diagnosis is for you to then follow in and either give it a cure or a treatment. So prenatal diagnosis is essentially the precursor for our end goal of designer babies. And designer babies just being children that have had their genetics modified in any way to either be more resistant to certain genetic diseases or just disease in general. It can even influence their traits. But initially this would start as just a way for us to eliminate genetic diseases through fixing chromosomal abnormalities. But eventually it would probably lead us to manipulating certain characteristics, traits, even personalities of our babies. So we have a couple tools at our disposal and actually able to accomplish this. Most notably, the 3 are pre-implantation diagnosis, which involves analyzing the female gametes and picking the most viable eggs determined based on lowering the risk of the targeted diseases. And we can even use this to better determine gender, or predetermine gender. We also have talons, which comprise of a non-specific DNA cleaving nucleus fused to a DNA binding domain that can be easily engineered so that talons can target essentially any sequence. The talons method is already being used to design plants, fuels, and also pets with certain specifications. And also we have the CRISPR-Cas9 system, which on the other hand requires a 3-base PAM sequence. So on the RNA lettering for the AUG or TAC, it basically wants a stop sequence of NGG immediately after the gRNA target sequence on the DNA. So basically in a super watered down and probably insultingly simplified explanation, Talons goes in and knocks out the RNA coding while CRISPR goes in, cuts, and then reprints, which may result in a much more robust cleavage. However, with CRISPR, it offers a much more widespread and efficient form of RNA cleavage as it's guided by the Cas9 enzyme, whereas Talons is a much more specific in its targetization, even if it is a little bit more clean at when it's cleaving the RNA sequence. So of these 3, only pre-implantation diagnosis has been used for human pregnancies. However, as our ability to diagnose prenatally becomes more exact, it really opens the doors for us to be able to begin using our precise DNA modification tools. So we essentially need to get better at just honing in on our locations before we can send in our laser-guided surgeons. But it's eventually we may be able to use prenatal diagnosis to be able to detect all or any specific chromosomal abnormality, and just with CRISPR just being the easiest to administer, we'd be able to mix up essentially kind of a CRISPR cocktail based on the diagnosis to then be injected into the fetal site, correcting any errors in the fetus's genome. So this, of course, is all speculation. With our current and constantly growing understanding of the human genome, we can isolate certain gRNA sequences that have positive correlations to either like longer lifespans, higher intelligence, disease resistance, eye color, hair color, muscle mass. The possibilities are endless. But in order to get to these end goal scenarios, we first need to continue down our path of perfecting the prenatal diagnosis based on the nucleated fetal cell analysis.